Okay, let's unpack this. Imagine holding the complete blueprint for a living organism. Every single instruction, neatly laid out. It sounds like science fiction, right?
Hmm.
But it's not. It's the incredible reality of genomics. And today, we're diving deep into how scientists actually create those intricate biological manuals. We're exploring genomics, the molecular analysis of an entire species genome. When we talk about a genome, we mean the complete genetic composition of an organism. Everything from a tiny bacterium with its single circular chromosome
Mm.
to humans with our, extensive set of chromosomes, and even a mitochondrial genome. Genomics is about taking that whole genetic blueprint and deciphering its components, understanding its structure, and, well, pinpointing every location.
Exactly. Our mission today is to uncover the fascinating strategies scientists use to map and sequence these entire genomes. You'll discover the ingenious experimental methods and the remarkable insights gained from these massive undertakings. It's about not just what genetic information is known, but how we came to know it, fundamentally reshaping our understanding of life itself.
What's truly remarkable about this field is how recently these comprehensive blueprints became accessible. Believe it or not, the first entire DNA sequence of a genome from the bacterium Haemophilus influenza was only completed in 1995. That was a team headed by J. Craig Venter and Hamilton Smith. Just 1 year later, the first eukaryote genome, Baker's yeast, followed. These were monumental steps that, well, they just blew open the doors to a whole new era of biological research.
And that ability to analyze whole genomes, not just individual genes, has absolutely revolutionized biology and medicine,
Yeah,
allowing us to see connections and patterns previously hidden.
absolutely. So when we talk about building this instruction manual, the first major step is often mapping. In genetics, mapping is the experimental process of determining the relative locations of genes or other segments of DNA on individual chromosomes. Think of it like drawing a detailed street map of a complex city you're exploring for the first time.
Right. And to continue that analogy, there are three general strategies scientists use to create these chromosome maps. Each one provides a different level of detail and serves, unique purposes: cytogenetic mapping, linkage mapping, and physical mapping. Cytogenetic mapping is your initial bird's eye view, so to speak.
Hmm.
It locates specific DNA sequences, like genes, within chromosomes that are actually visible under a microscope. These chromosomes often display characteristic banding patterns when stained, right? And genes are mapped relative to these visible landmarks.
Okay, so this method is commonly used for eukaryotes with their larger chromosomes. For example, the human CFTR gene, which, when defective, causes cystic fibrosis, is located precisely on chromosome 7 in the Q3 region. While incredibly useful for that broad localization, cytogenetic mapping typically offers a resolution of around, 5 million base pairs? Though you said it can be much finer in species like Drosophila with their specialized polytene chromosomes.
Exactly. And to achieve this microscopic precision, a key technique is fluorescence in situ hybridization, or FISH. As the in situ implies, the procedure is conducted directly in place on chromosomes held on a slide. So cells are prepared, their DNA strands separated or denatured, and then a specific DNA probe complementary to the target gene is introduced. This probe is tagged, usually with something like biotin, so it can be detected. Then you add something fluorescently labeled, like avidin, which binds the biotin. This lets scientists see exactly where that gene is located on the chromosome under a fluorescence microscope, often using a counterstain to see the rest of the chromosome structure and relating it to those banding patterns.
Ah, okay. And a visually striking application of FISH is chromosome painting. So imagine using multiple different probes, each labeled with a different fluorescent color, to literally paint different sites along a single chromosome in distinct hues. It's like creating a vibrant, color-coded map of your DNA. Very cool visually.
It really is. And what's fascinating here is that CyTish isn't just for basic research, it's also incredibly valuable in clinical settings. Clinicians use FI to quickly detect significant changes in chromosome structure, things like deletions, duplications, or translocations. These structural changes are often associated with genetic disorders, right? And FIs can reveal them with speed and precision. This helps diagnose conditions or distinguish, for instance, between a large gene deletion and a subtle point mutation in a disease like, say, phenylketonuria or PKU.
Okay, so if cytogenetic mapping provides the broad strokes, linkage mapping really drills down into how often genetic information shuffles around. This approach uses the frequency of genetic recombination or crossing over between different genes to determine their relative spacing and order along a chromosome. Distances are expressed in map units, which are also known as centimorgans.
Right. And historically, linkage mapping relied on observable traits caused by specific gene alleles, like Mendel's P's, essentially. But the game changed dramatically with the introduction of molecular markers. These are segments of DNA found at specific sites that can be uniquely recognized using molecular tools like PCR and gels, regardless of whether they actually encode a gene or not.
So what exactly made these molecular markers such a game changer compared to, just looking at traits?
Well, their power lies in their polymorphism. They vary significantly from individual to individual within a population, and their sheer abundance. They provide thousands of distinct reference points across the genome, making it much, much easier to identify connections than tracking traditional traits. There are many types: restriction fragment length polymorphisms, RFLPs, amplified restriction fragment length polymorphisms, AFLPs, microsatellites. Microsatellites are also called short tandem repeats, or STRs. You might have heard of those. And single nucleotide polymorphisms, SNPs, all leverage subtle variations in DNA sequences. But let's zoom in on microsatellites for a moment. They're a particularly clear example of how these markers work.
Right, microsatellites. Those are short, repetitive DNA sequences, like the CAN dinucleotide repeat in humans, where N can vary in length. What makes them so useful is that these length variations are highly individual, very polymorphic. So researchers design PCR primers that flank these unique repetitive regions, amplify them, and then use gel electrophoresis to detect the exact length of the amplified fragment for each individual. A longer fragment means more repeats, a shorter one means fewer. Simple as that, really.
Exactly. And this leads us to a crucial application: tracing disease-causing alleles in humans, which is, a fascinating challenge. If we can't perform experimental crosses like we do with fruit flies or plants, how do we actually track these genetic locations in humans?
Yeah, that's a good question. How do we pinpoint a disease-causing gene in a family?
Well, the answer lies in analyzing human pedigrees and leveraging the concept of a founder. We assume a disease-causing allele originated in a single individual, the founder, generations ago. Now, if a polymorphic molecular marker is physically very close to that disease-causing allele on the chromosome, it's highly unlikely that a crossover event will separate them during meiosis, right? They're linked. Therefore, you can track the transmission of this closely linked polymorphic marker through a large family pedigree. If the marker consistently appears with the disease across generations, it provides a strong clue that the marker and the disease gene are linked. That gives you a powerful way to narrow down the gene's location.
Okay, that makes sense. So from the relative distances of linkage mapping, we move to physical mapping, which takes us to actual measurable distances. This third approach uses DNA cloning and sequencing to determine not just the relative order, but the exact locations and distances between genes and other DNA regions, measured precisely in base pairs. It's like getting exact GPS coordinates rather than just south of the park, much more precise.
Exactly. And the central concept here is the contig. That's a collection of overlapping DNA clones that, when assembled, form a continuous stretch of DNA. That represents a physical map of a chromosomal region. Researchers build these by identifying shared sequences between cloned DNA fragments, piecing them together like an intricate biological puzzle.
Right, and to build these contigs, especially for large eukaryotic genomes, you need specialized cloning vectors, things that can accommodate very long DNA inserts. This led to the development of artificial chromosomes. The yeast artificial chromosome, or YAC, developed back in 1987, was groundbreaking. It was capable of carrying inserts up to 2 million base pairs.
Huge fragments.
Yeah. Even more commonly used today are bacterial artificial chromosomes, or BACs, and P1 artificial chromosomes, PICCs. These typically carry inserts up to maybe 300,000 base pairs. They're apparently a bit easier to work with than YACs. And for even smaller inserts, maybe tens of thousands of base pairs, there are cosmids. Using these allows scientists to efficiently build these large overlapping collections, creating those precise physical maps.
And physical mapping directly aids in what's called positional cloning. This is where a gene is cloned purely based on its mapped position on a chromosome. One powerful technique here is chromosome walking. Sounds quite descriptive, doesn't it? To start, you begin from a known gene or marker, which serves as your anchor point on the map.
Okay, so you have a starting point. How do you walk?
From that anchor, researchers take methodical steps. They subclone a small piece of DNA from the very end of one clone, the part furthest from the known starting gene. This little piece is then used as a probe to find the next overlapping clone in a library, the one that extends further into the region of interest. You repeat this subcloning and screening process, taking step after step, literally walking along the chromosome toward your gene of interest.
Wow, that sounds painstaking. But this technique has been incredibly impactful, right? It was successfully used to clone genes for significant human genetic diseases like cystic fibrosis, Huntington disease, and Duchenne muscular dystrophy. It's a real testament to molecular ingenuity, like reverse engineering a complex mechanism without a manual, just one tiny piece at a time.
Absolutely. And the crucial question becomes, how do you know when you finally reach the target gene? You've been walking, but where's the destination? Well, if you're looking for a disease-causing gene, you conduct your chromosome walk using DNA from both affected and unaffected individuals. When you reach a point where the DNA sequences consistently differ between them, say a mutation present only in the affected individuals, that difference is very likely within your target gene. You've arrived.
Okay, so mapping is about understanding the layout and relative distances. It sets the stage. But the ultimate goal of all these efforts is to achieve a full readout, determining the complete DNA sequence of an entire species genome. This is where large-scale genome sequencing projects come in, the big picture.
Right. And the primary approach for large-scale sequencing is called the shotgun method. Genomic DNA is first isolated and then randomly broken into smaller fragments, lots of them. Each of these fragments is then sequenced individually. And the real magic, or maybe the heavy lifting, happens with bioinformatics tools. These tools identify overlapping regions between these randomly sequenced pieces, much like assembling a giant puzzle without the picture on the box. This allows them to reconstruct the complete sequence of the entire genome.
The big advantage of shotgun sequencing is its efficiency, isn't it? It doesn't require extensive time-consuming prior mapping. While you do inevitably sequence some regions multiple times, which might seem redundant, it's remarkably effective. For the Haemophilus influenza genome, for example, which is about 1.8 million base pairs, they sequenced 9 million base pairs. That's 5 times its length. And even then, it still left only about 0.67% of the genome unsequenced. That's an incredible hit rate for a random process.
It really is. That 1995 H. influenza project by Venter, Smith, and their colleagues truly was a landmark. It revealed the first comprehensive genome picture of a free-living organism with its 1.83 million base pairs, and they predicted about 1743 genes from that sequence.
And this is where the sheer scale of these projects truly hits home. The Human Genome Project, for instance. That was the largest biological project of its kind, a monumental 13-year international effort. Ran from 1990 to 2003, roughly, with the final maps and sequences published a bit later by 2006. It was coordinated by the U.S. Department of Energy and the NIH, and it aimed to sequence our entire 3 billion base pair genome. To put that in perspective, if our genome's base sequence were typed into a textbook, it would be nearly 1 million pages long. Just immense.
Yeah, the Human Genome Project had incredibly ambitious goals. Beyond simply obtaining the DNA sequence with its first draft published in 2001 and completion by 2006, it also aimed to develop the technology for managing and analyzing the staggering amount of genomic information. Bioinformatics became huge. They also focused on analyzing the genomes of crucial model organisms like E. coli, yeast, fruit flies, mice. Understanding them helps us understand ourselves. And critically, they established programs to address the ethical, legal, and social implications, the ELSI issues of genomic information. Thinking about things like privacy and potential discrimination, that was built in from the start.
That's so important. And the profound impact of this project cannot be overstated. It didn't just give us a sequence, it gave us the index to a vast biological library, you could say. This immediately transformed disease research, allowing us to rapidly pinpoint genetic predispositions and develop targeted therapies, things that were unimaginable just a few decades ago. It's essentially shifting medicine from a one-size-fits-all approach to one that's increasingly personalized.
Yes, absolutely. And the work didn't stop there. In 2008, the 1000 Genomes Project was launched. That established an even more detailed understanding of human genetic variation by sequencing thousands more genomes from around the globe. It provided critical insights into human diversity and susceptibility to various diseases.
What's truly remarkable here, though, is how innovations in DNA sequencing have made it dramatically faster and cheaper. The Human Genome Project was initially estimated to cost around $3 billion for one human genome. By 2019, the cost could be less than $1,000. That's just an astonishing drop. This innovation is rapidly making it feasible to sequence your genome as a routine diagnostic procedure, ushering in this new era of personalized medicine we talked about.
Yeah, these dramatic cost reductions are thanks to high-throughput and next-generation sequencing technologies, or NGS. These involve massive automation and parallel sequencing, meaning they can perform thousands or even millions of sequence reads simultaneously, not just, 96 at a time like older methods. Critically, these next-generation technologies largely supersede older methods like Sanger sequencing for whole genomes by often eliminating the need for time-consuming DNA cloning steps. That saves immense time and money.
Can you give us an example of one of these next-gen methods? How do they achieve that speed?
Sure, let's take pyrosequencing as an example. It's a type of sequencing by synthesis, or SBS.
Okay, sequencing by synthesis.
Right. Imagine detecting light signals every time a DNA building block, a nucleotide, is added to a growing strand. Pyrosequencing relies on a cascade of enzymatic reactions. When the correct nucleotide is incorporated by DNA polymerase, it releases a molecule called pyrophosphate. This pyrophosphate triggers further reactions involving enzymes like ATP sulfurylase and luciferase, yes, the enzyme that makes fireflies glow. This ultimately produces a flash of light. This light is detected by a sensitive camera, allowing real-time monitoring of which nucleotide was added at each step. It's quite ingenious.
Wow, detecting light flashes. That's amazing.
It is, and if we connect this to the bigger picture, the astounding advances in DNA sequencing technology have led to an explosion in the number of sequenced genomes. By 2018, over 5,000 prokaryotic and 300 eukaryotic genomes had been sequenced, and that number is way higher now. The motivations are incredibly diverse, from basic research on model organisms to understanding human diseases by sequencing pathogens, improving agriculture, and even for comparative genomics to understand evolutionary relationships between species.
Okay, so we can sequence individual organisms. But now imagine if most of the world's life forms couldn't actually be studied in a lab. That's the reality. For the vast majority of microorganisms in environments like soil, water, or even your own gut, they simply cannot be cultured traditionally. This profound limitation led to the development of metagenomics, a truly groundbreaking field.
Yeah, metagenomics is defined as the study of a complex mixture of genetic material, what we call a metagenome, obtained directly from an environmental sample. It completely bypasses the need to culture individual organisms in a lab. This allows us to unlock the genetic secrets of this vast, unseen majority of life.
So how does that work practically? What's the strategy?
Well, the strategy is brilliantly straightforward in concept, though complex in practice. You obtain an environmental sample, say a scoop of soil, a liter of ocean water, maybe a gut sample. You then sterilize all the cells in that sample, extract and purify all the DNA present from potentially thousands of different species. Then you shear this mixed DNA into fragments. You can either insert those fragments into cloning vectors to create what's called a metagenomic library, or increasingly, you just use next-generation sequencing technologies to directly shotgun sequence this entire mixture of DNA.
So you're sequencing everything that's in there all mixed together.
Exactly. And the applications of metagenomics are incredibly diverse and impactful. It's not just about finding new microbes, it's revealing entire unseen ecosystems. These ecosystems drive planetary processes, from nutrient cycles to climate regulation. It's forcing us to fundamentally reevaluate life's true diversity on Earth. In human medicine, it's revolutionizing our understanding of the microbiome, those complex microbial populations in our bodies, and their role in health and disease. For biotechnology, it's a goldmine for discovering new enzymes, new chemicals like novel antibiotics made by previously unculturable microbes.
That sounds powerful. Can you give us a specific example?
Absolutely. Here's a truly fascinating one. J. Craig Venter's ambitious global ocean sampling expedition. Remember him from the first bacterial genome?
Right, the H influenza sequence.
Well, in 2003, his 95-foot sailboat, the Sorcerer II, was outfitted as a research vessel. It embarked on this incredible 32,000-mile journey, collecting water samples every 200 miles or so across the Atlantic and Pacific Oceans. They filtered out the microbes and performed shotgun sequencing on the DNA. This yielded an unbelievable 7.7 million sequencing runs, totaling over 6 billion base pairs of DNA data from just ocean water microbes.
Wow, 6 billion base pairs. What did they find in all that data?
The discoveries were truly surprising. The expedition found hundreds of previously undiscovered genes. For example, genes for a light harvesting protein called proteorhobson, which was thought to be rare but turned out to be common. Even more astonishing, they identified over 1,800 new species of microorganisms just from those samples, dramatically expanding our known tree of life.
1,800 new species.
Yeah.
Just like that. And what about this paradox they uncovered?
Ah, yes, the paradox. They found the coexistence of many, many closely related species and subtypes thriving together in the same bucket of water, essentially. This challenged previous ecological assumptions about competitive exclusion, the idea that one species should dominate and outcompete similar ones in the same niche. How do these diverse but very similar species manage to thrive together without one taking over?
That's a really interesting question. Did they figure it out?
Well, it raised that profound question, and it's one that continues to drive ecological and microbial research today. It suggests a level of microbial specialization, maybe subtle differences in resource use or environmental tolerance that we're only just beginning to understand, a much more complex web of interactions than previously thought.
Okay, so we've covered a lot of ground, from tracing a single gene on a chromosome using mapping techniques, all the way to decoding the entire genetic makeup of diverse ecosystems with metagenomics. We've seen how these deep dives into DNA are constantly reshaping our understanding of life itself. We talked about the meticulous art of mapping cytogenetic linkage, physical mapping with contigs, and chromosome walking. And we covered the monumental genome sequencing projects, like the Human Genome Project and the incredible speed and affordability brought by next-generation sequencing. Finally, we peeked into that unseen world through metagenomics, unlocking the secrets of unculturable life forms. It's really an amazing journey.
It really is. So what does all this mean, big picture? I think the more we discover about the genetic blueprints of life, from the simplest bacteria to complex ecosystems like the ocean microbiome, the more we realize how interconnected and truly intricate life is. Every new deep dive into a genome reveals new secrets, constantly reshaping our understanding of biology and, our own place within it. What profound connections, or perhaps even entirely new branches of the tree of life, will the next sequence reveal? It makes you wonder.
It certainly does. Thank you for joining us for this deep dive into the fascinating world of genomics. We hope it has sparked your curiosity and left you feeling truly well-informed. Keep exploring, keep questioning, and keep digging deeper.